Good afternoon, dear students. Yesterday we discussed about inducible and repressible system of control of gene expression or lac operon control by inducible and repressible system. Today we are going to discuss about positive and negative control of lac operon. Right. What is negative control? In the lac operon model that is proposed by Jacob and Monod, the presence of regulator protein, the presence of regulator protein inhibits the expression of structural genes. Thus, the regulator protein negatively regulates the expression of lac operon. Okay, so gene whose expression is switched off due to the presence of regulator protein are said to be under negative control. Okay, we can see that yesterday we discussed that in both inducible system as well as in repressible system, protein synthesis takes place only when the operator gene is free. Such a control mechanism for protein synthesis is thus of negative type. Whatever we discussed yesterday, whether it is inducible system or repressible system of operon control, in both the cases the operon is under negative control because the operon expression, that is synthesis of enzymes by structural genes, takes place only when operator gene is free. Okay. So example of negative control is induction of protein synthesis in the presence of lactose. As soon as lactose becomes available, or in the presence of lactose and in the absence of glucose, lac operon is expressed and protein synthesis take place. Okay. In negative control, the regulator protein is the repressor. Okay, or rather we can say that repressor is a protein synthesized by the regulator gene, and the repressor prevents gene transcription. Okay, the controlling site is the operator gene of the lac operon. In inducible system, the effector molecule is the inducer. The inducer prevents the repressor from binding the operator and thus allows gene transcription or protein synthesis to take place. In the repressible system, the effector molecule is a co-repressor. Co-repressor binds to the aporepressor. Then together they two gets activated and then they bind the operator gene and prevent gene transcription. Okay. So in inducible system, the effector molecule is inducer, which allows expression of operon. In repressible system, the effector molecule is a co-repressor which prevents gene expression. That means which prevents enzyme synthesis. So in both the cases, the operon is under negative control. Okay, so you can look at this diagram. The regulator gene has synthesized a active repressor protein which binds the operator, blocks it, and the system is repressed. No protein synthesis. Okay, as soon as inducer becomes available, the inducer inactivates the repressor. Okay, you can see here the inducer inactivates the repressor and hence system is induced. That means lac operon is switched on. Okay, so here we can see that the repressor has bound and system is off, and inducer inactivates the repressor and system becomes on. Okay, what happens in repressible system? In repressible system, the regulator gene synthesizes inactive repressor. Okay, so inactive repressor cannot block the operator and hence protein synthesis takes place. Okay, as soon as co-repressor becomes available, it activates the repressor and the structural genes are repressed. That means protein synthesis is stopped. Okay, this is about repressible system. Okay, what is positive operon control? A qualitatively different regulatory system also exists in prokaryotic cell. This system is a positively controlled system. Here the presence of regulator protein switches the system on. Okay, so gene whose expression is switched on due to the presence of regulator protein are said to be under positive control. In positive control, regulator protein acts as an activator and increases enzyme synthesis. the activator attaches to a separate controlling site on the DNA. The initiation site or the binding site of activator is not the operator. Okay. Since regulator protein enhance or increase or allows gene transcription, this type of regulation is called as positive regulation of protein synthesis. Okay, so sometimes the regulator gene synthesizes inactive activator. This inactive activator is activated by inducer molecule, which then binds to the operon and switches the system on. Sometimes the regulator gene synthesizes active activator protein, which can directly bind the operon and turn the system on. The system will remain on. On means what? Protein synthesis or enzyme synthesis from structural genes will continuously go on. Okay. So the system will remain on till the effector molecule becomes present in the cell. The effector molecule then binds the activator protein, makes it inactive, thereby shutting the system off. The effector molecule here is a co-repressor. Is that clear? So, inducer activates inactive activator. Co-repressor inactivates the active activator protein. Right? So, whether the system should remain on or off is under the control of effector molecules. Right. Now, we can see in this diagram the first two. Okay. First of all, the regulator gene produces an inactive activator protein. Okay, the inactive activator protein, if it is inactive, it cannot bind the initiation site and hence system is off. Protein synthesis is not taking place. As soon as inducer becomes present, it activates the activator. Okay, so inducer binds to the inactive activator, it converts it into active activator, and the active activator binds to the initiation site and allows movement of RNA polymerase and protein synthesis takes place. Okay, this is about inactive activator synthesized by regulator gene. The inactive activator is activated by inducer molecule, which makes the system on. Protein synthesis starts. Okay. Now let us discuss what happens when the regulator gene produces active activator. Okay, here in this case, the regulator gene has produced active activator molecule. So it is bound to the operon. It is bound to the operon and hence RNA polymerase carries out its work and system is on. Operon expression is taking place. Enzyme synthesis is taking place. But as soon as co-repressor comes into the environment, co-repressor converts active activator into inactive activator. So as soon as the activator becomes inactive, it cannot bind to the operon. Hence, the system becomes off. Protein synthesis is shut down. Okay, this is called as positive operon control. Now let us discuss another point, that is what is the role of cyclic AMP in regulation of lac operon expression. Okay. Now early indications or early experimentation proved by Earl Sutherland proved that cyclic AMP is involved in the catabolic repression. Okay, cyclic AMP is involved. Cyclic AMP is involved in catabolic repression. What Earl Sutherland demonstrated that glucose lowered, okay, presence of glucose lowered the intracellular concentration of cyclic AMP from 0.1 millimolar to 0.1 micromoles. Okay, even more directly, Zube proved that cyclic AMP increased the synthesis of beta-galactosidase enzyme 30 folds. Okay, so presence of cyclic AMP increases the synthesis of beta-galactosidase. So cyclic AMP has a direct role in increasing protein synthesis, enzyme synthesis. Okay. Cyclic AMP is thus involved in the catabolic repression, and the concentration of cyclic AMP is connected to the concentration of glucose. Simply we can say that When the concentration of glucose is high, the concentration of cyclic AMP is low, or vice versa. But glucose directly does not control the level of cyclic AMP. A metabolite of glucose represses the synthesis of cyclic AMP. How is cyclic AMP produced? It is produced from ATP by the action of enzyme adenyl cyclase, which is the protein product of the gene CYA. Okay. Now, bacterial cells contain a protein called cyclic AMP receptor protein. It is called as CRP, which was previously called as CAP, catabolite activator protein. This protein is produced by CRP gene. The protein is the positive control factor whose presence is necessary to initiate transcription of lac operon and other operons whose promoters are dependent on CRP. So rather, simply saying, the promoter, which is the binding site of RNA polymerase, is actually dependent upon CRP. What is CRP? Cyclic AMP receptor protein. So CRP protein is activated only when it is bound to cyclic AMP. That means the concentration of cyclic AMP should be high in the cell so that it can bind CRP, activate the CRP, then the active or cyclic AMP bound CRP will go and bind the promoter region, and then it will initiate RNA polymerase binding, and hence structural genes will be expressed and enzyme synthesis will take place. So cyclic AMP plays a direct role, or it is directly involved in increasing the enzyme synthesis of lac operon. But what happens when glucose concentration is high in the cell, or when the glucose concentration is high in the medium? Organism utilizes glucose, and hence at that time cyclic AMP concentration is very low. When the concentration of cyclic AMP is low, CRP is inactive because cyclic AMP is not bound to it. The inactive CRP cannot bind the control region of lac operon, and hence in its absence, RNA polymerase cannot initiate transcription. That means RNA polymerase needs to be pushed, need to be activated to initiate or to start mRNA and protein synthesis. That is done only by CRP bound to cyclic AMP. Okay. On the other hand, when glucose concentration goes down, that means glucose concentration becomes low, cyclic AMP level shoots up. Cyclic AMP concentration goes on increasing. As the cyclic AMP concentration increases, it now binds to the CRP protein. The cyclic AMP CRP bound complex is active. It binds to the control region on the operon, activates RNA polymerase enzyme, which will then start protein synthesis, or operon expression will take place. So look at this, right? What happens when glucose is present? Cyclic AMP is low. Okay, that means glucose is present, cyclic AMP is low, and lactose is absent. That means operon will not be expressed. No mRNA and no protein synthesis. Now when glucose is present, cyclic AMP is low, lactose is also present. But then the synthesis of proteins will be very slow, or they won't be synthesized. What happens when glucose is absent? When glucose is absent, cyclic AMP concentration is high. But if lactose is absent, mRNA synthesis or protein synthesis will not take place. Q. Why? Because if lactose is absent, the enzymes like beta-galactosidase are not needed. Okay. Now when glucose is absent, cyclic AMP concentration is very high and lactose is present. Then rapid mRNA synthesis or rapid synthesis of enzymes like beta-galactosidase, permease and transacetylase will take place. Okay. So no glucose, high concentration of lactose, that means high concentration of cyclic AMP, and rapid protein synthesis will take place. Okay. So the above studies indicate that lactose must be present and glucose must be absent simultaneously for full induction of expression of lac operon. Okay, in order that the lac operon expresses to its fullest and produces all the three enzymes in large quantity, it is necessary that lactose should be present and glucose must be absent. This means that both positive and negative regulation must occur simultaneously, and hence we say that lac operon is both positively as well as negatively regulated. Okay, now look at this simple diagram. When the glucose concentration is high, okay, when glucose concentration is high, what will happen? Low cyclic AMP concentration. And when cyclic AMP concentration is low, the CRP is inactive. Okay, the CRP is inactive. If the CRP is inactive, it cannot bind the lac operon, and hence no transcription will take place. Okay, so is that clear? No transcription when glucose concentration is high. What will happen if the glucose concentration is low? Okay, so low glucose concentration, high cyclic AMP concentration converts or active cyclic AMP CRP complex. This active complex binds the lac operon and transcription takes place and protein synthesis will take place. Okay. I hope you understood. So glucose concentration low, high cyclic AMP concentration, it will bind the CRP and convert it, make it active. So active cyclic AMP CRP complex, it binds the operon and transcription takes place. Okay, so cyclic AMP has a direct role in expression of lac operon. So if cyclic AMP is present in high concentration, it will activate the CRP protein, which will bind the lac operon and which will activate RNA polymerase enzyme, and protein synthesis will take place. So dear students, that is all about lac operon. So we have discussed the structure of lac operon, the promoter, regulator, operator gene, what are their functions, the structural genes, what are the three structural genes, that is Z, Y, A, what enzymes they synthesize, all we have discussed. We have also discussed about positive gene regulation, negative gene regulation, etc. Okay, so that is all about lac operon.